So, after this long, irrelevant rant, let's talk about digestion and absorption. And I kind of put these two things together, okay? Because together, they make sense. So we're going to start with the title, okay? So we are at the moment when Kim is in the small intestine, duodenum or in the jejunum, ileum. So you've got a whole bunch of biological molecules, and the first biological molecule that I'm going to talk about will be proteins, right? So proteins that consist of amino acids are broken down to peptides by the enzymes in the intestine. So it will be pancreatic enzymes. And also if you remember the long and convoluted path that food has taken so far, proteins are also broken down by pepsin in the stomach. Does that make sense? So at this point, you went from massive proteins to much smaller peptides. And then, drum roll, the enzymes that we so far ignored, enzymes found in the brush border, okay, of the columnar epithelium in the intestine. So these enzymes, the enzymes that I'm going to talk about, they are found in those microvilli. There are two enzymes. It's Aminopeptidase and Dipeptidase. And they will chew up peptides to amino acids. Okay, makes sense so far? So you're progressively breaking down large biological molecule, macromolecule, into smaller, into individual building blocks. And that happens, pancreatic enzymes, brush border enzymes. We good? Now, here I'm going to put polysaccharides. Okay. I want to highlight the similarity between large proteins and large polysaccharides. So polysaccharides will be broken down to smaller chunks by the pancreatic and salivary amylases. Into the oligosaccharides. In the lecture notes, there is a table with the brush border enzymes, and it lists several brush border enzymes. They break down oligosaccharides. For instance, let me exemplify. Listen. You eat a starchy food. Starch is the polysaccharide. Does that make sense? Amylases break down starch to maltose, dextrose, maltriose. Okay, does that make sense? These oligosaccharides are then digested to monosaccharides by brush border enzymes. Are we clear? So again, you go from very large molecule to smaller molecule to elementary building blocks. Does that make sense? What do you need to know? I can ask you, what is the function of such enzyme? What is the function of, for instance, lactase? To break down lactose. Sucrase, break down sucrose. Make sense? Maltase, break down maltose. Amylase, break down starch. This is the memorization part. So there are two tables, big tables in the notes. One with brush border enzymes, one with the pancreatic enzymes. Be able to tell me what these enzymes break down and more or less into what. I am more focusing on what they break down rather than what the products are. Does that make sense? Go ahead.
The pancreatic enzymes and the brush border enzymes are both in the intestine.
Yes.
But the pancreatic enzymes hit the proteins or whatever, like the nutrients first.
Yes. Moreover, brush border enzymes are physically attached to the membrane. Like, they are not just swimming in the brush border. They're sitting on the membrane. While pancreatic enzymes are in the lumen, like they are released as the part of the pancreatic juice. They kind of not bound, they free. Does that make sense? So this part, does that make sense to you so far? Okay. Next idea that we're going to talk about will be how do we absorb these nutrients, monosaccharides and amino acids. And to answer this question, I'm going to draw schematics here. So check this out. This is the layer of epithelial cells in the intestine. Okay? Does that make sense? Now, what is the ultimate goal? What is the ultimate goal of absorption? Ultimate goal is, look at me, to move amino acids or monosaccharides or whatever nutrient we're talking about from the lumen of intestine here into the blood. Does that make sense? So ultimately, you need to, these nutrients have to cross the epithelium and get into the blood. Are we clear? Good? How do we do it? We're going to start with a quite unexpected story. We're going to start with the basal side of the epithelium right here at the bottom. Can you all see that? I can't really do anything about the computer because it will compromise my recordings. So basal side, all right, the one that faces blood. Can you see that? Turns out on the basal side of the epithelium, there is a sodium-potassium pump. Now, follow my logic. I'm going to tell you what happens. You're going to tell me the outcomes. So sodium-potassium pump constantly pumps sodium out of the epithelial cells. Does that make sense? So since sodium is permanently, not permanently, but continuously pumped out of the cell, what happens? What's the concentration of sodium inside the cell? Is it high or is it low if the sodium is constantly pumped out? Pumped out. Exactly, because think of it. You're pumping sodium, you're getting sodium out of the cell. Does that make sense? So we have very little sodium inside. Very little sodium inside. Like very, very little. What's important is this. Check this out. concentration of sodium in the lumen is higher. Okay? How do we call a situation when there is a, concentration is higher and then it becomes lower? What do we call it? The difference. The G word, the gradient, yes. We have a gradient of sodium concentration. Does that make sense? So sodium just will diffuse. That's your sodium moving along the concentration gradient. So sodium will diffuse through the, apical side of the epithelium into the cell. Am I clear? I'm going to check how attentive you are. I will ask you what happens to that sodium once it's inside the cell. It gets pumped out. Does that make sense? So this pump creates a flow. You see what I'm saying? Does that make sense? This pump creates a flow of sodium. So sodium can diffuse here. Are we clear? Why it's important? Neither amino acids nor monosaccharides either of them can't diffuse into the cell. I kind of got lost in the pronouns, okay? So these and these can't diffuse into the cell because there's no gradient. See what I'm saying? So what they use instead is so-called secondary active transport. The diffusion of sodium, now think about it as like a ball rolling down the slope. Are you following me? If at the bottom of the slope there is another ball, this one will hit it. Make sense? So the second one will go up against the concentration gradient. Does that make sense? So, what happens? Diffusion of sodium, for instance here, diffusion of sodium will provide energy for basically pumping monosaccharides into the cell. Diffusion of sodium will provide energy for pumping amino acids into the cell. Does that make sense? So this, for instance, this molecule is called sodium amino acid symporter. Is that clear? Now I want to highlight something here, kind of go over it once again. So this digestive part, I hope, is clear, right? So due to sodium-potassium pump, on the basal side, concentration of sodium inside the epithelium is low. That allows sodium to diffuse into the epithelial cells, and the energy of that diffusion is used to pump monosaccharides and amino acids into epithelial cells. Does that make sense? And then on the basal side, amino acids and monosaccharides use facilitated diffusion. Just transporters, just some channels or carriers at the bottom. So if we want to describe the transport of sodium, what do we see? We see active transport of sodium is the basal side, and it drives the diffusion on the apical side. Diffusion on the apical side drives secondary active transport of amino acids and monosaccharides, which then use facilitated diffusion to go here. Does that make sense? Good? Questions?
Is this sodium? is happening even if there's no amino acid?
Yes, all the time. All the time. A huge chunk of energy that we use for our day-to-day lives is just to pump sodium all the time. Activity is regulated by the concentration, okay? It's not like, it's going to pump sodium until cell is like bleeding dry, It's the balance. But the point is, this pump is essential to maintain that low concentration, which allows for sodium to diffuse. When you eat food, it contains sodium. So if you will somehow, completely, which is impossible, strip your food from sodium, like at all, like nothing, again, it's impossible. If you would do that, technically, you would have no absorption. Does that make sense? The idea here. Okay? Got it? Again, you just have to think like in stepwise here. Now, while we're at it, I'm going to put fats on the back burner for now, and I want to discuss. Can I kill the nutrients? Okay. So I want to discuss the absorption of some ions. What follows sodium? There you go. So sodium crosses the epithelium. Does that make sense? We just drew it, right? Sodium crosses the epithelium. It diffuses into the cell and then is pumped. And we can put like water follows sodium by osmosis. Are we clear? Now, let's not, kid ourselves. It's pretty boring. Come on, I know. It is. No, you work night shifts. I have no, you yawning. No, it's fine. We humans, that's what humans do. That's our physiology. I would be surprised if you wouldn't. But it's boring. Like seriously, it is. I know. Now here's the thing. What's going to happen if, for some reason, sodium absorption, whether you have a problem with a sodium-potassium pump, or you have a problem with a channel here, okay? What if the sodium absorption is impaired and sodium is not being absorbed from the lumen and it stays in the lumen? What's going to happen? So what you're going to have? No. Well, eventually, yes. Eventually you're going to have dehydration. But you're not like running to a physician saying, "Doctor, I have dehydration." What you're going to have? When water stays in your intestine excessively. Huh?
Swelling.
Diarrhea. Simply diarrhea. You have watery diarrhea. Does that make sense? So basically, why, and that's kind of, it's really nice to explain physiologically. Why, for instance, when we have some bacterial infection in the stomach, why do we have, in the intestine, why do we have diarrhea? Because those absorptive structures, those microvilli, you see this? They're gone. When they're gone, they misshaped, so to say. When they're gone, sodium absorption is impaired. Sodium stays in the intestine, as does water, and you have watery diarrhea. That's all. Does that make sense? Just like understanding the basic stuff. Okay? Good? And another sort of, I'm kind of elaborating, but what do you do when you have watery diarrhea? Hopefully not due to the infection, but some maybe food poisoning. What do you have? What do you do? Huh? Take Pepto. That has slightly different gist. You can take Imodium, right? Another antidiarrheal drug. So what this drug does, it inhibits the motility of the intestine. Does that make sense? How muscles, move around, right? Make sense? So if motility and peristalsis are inhibited, what happens to the time that food, including sodium, spend in your intestine? It increases, right? If food moves slower. Does that make sense? Which means you have more time for proper sodium absorption. This is how diarrhea is treated. The food moves slower and it has time for absorbing water and then sodium and water and all that kind of stuff. That makes sense. One more ion to talk about here. Potassium. Now let's look at this picture that we have. So sodium gets absorbed, right? And water follows sodium. Now let me ask you this. When water is being absorbed from your intestine, now listen closely, how does water absorption change the concentration of other ions in here? When water is absorbed from the intestine, how does it change the concentration of other ions in here? Increases or decreases? Increases. Does that make sense? Because you take out water, you're concentrating. Make sense, Emma, Vicky? Okay. Which means potassium concentration goes up, and potassium diffuses, interestingly, primarily between the cells. Okay? Between the cells. So-called transcellular absorption of potassium. So those are three non-nutritional components that I want you to understand here. Am I clear? We good? So three things that are not nutrients. Like really mechanistically. So I can ask, what is the driving force for water absorption? Absorption of sodium through the epithelium, all right? Or how is water absorbed? By osmosis. What happens to potassium concentrations when water absorbs? Potassium concentration in the lumen increases, which allows for potassium absorption. Clear? Okay. Two ions, calcium and iron. So calcium. What regulates absorption of calcium? Vitamin D/parathyroid hormone. Okay. So that puts a big no on claims from our parents. If you drink more milk, your bones are going to be stronger. No. The bones are going to be as strong as they need to be. All excessive calcium that you consume, you will either just pass with feces or pee out. Does that make sense? So calcium is absorbed when there is need. Same goes for iron. Does that make sense? So when iron is absorbed, okay, let's be frank. For an otherwise healthy adult male, does it make sense for me to take iron supplements? No, because iron is absorbed when needed. I'm not going to boost my athletic performance by consuming iron because I'm not going to produce more red blood cells. Does that make sense? For a female, is there a reason to take iron supplements? Yeah, once a month. During menses, yes, because there's blood loss, there is associated anemia. Yeah, it makes sense. Clear? Okay.